Thanks. Okay, our next speaker is Pierre Joubert from University of California, Berkeley.
Oh, there's a timer there.
I'm going to restart it.
Hi. Okay. Hi, everyone. Thanks for coming, and thanks so much to the organizers for inviting me. I'm really excited to talk about this kind of interdisciplinary project that I've been working on for the past few years. So I'll be talking about, let's see. Clicking? Okay. So I'll be talking about the rice blast pathogen. I can take my mask off. Magnaportha oryzae today, as well as extrachromosomal circular DNAs and how I think the two might be linked. So like I said, Magnaportha oryzae is the rice blast fungus. Obviously, rice is a super important crop, feeds half the world, and rice blast eats about 30% of the crop every year. So you can see how this could be a really important model pathogen. Just give it a second. Yep. So one of the problems with this fungus is that it can evolve very quickly. One of the solutions that people are the most excited about for how to prevent this fungus from eating so much of our crops is to use genetically modified rice plants. The problem is that when those rice plants are deployed, rice blast is able to evolve and overcome that resistance very quickly, often in just a couple years. So I'm interested in figuring out how it's actually able to do that. So just a little bit of background on fungal pathogens for those of you that are less familiar with them. So fungal pathogens secrete effectors, sometimes into the plant cell, sometimes into the apoplast. But basically the idea is that, these effectors are going to target essential plant functions and then take over the cell in order to cause disease. So plants have immune receptors that detect some of these effectors, not all of them. And the idea is that there can be a lot of redundancy in these effectors. And so there's pressure for the fungus to lose or mutate some of those effectors so that the plant immune receptors can't detect those anymore. So the consequence of that is that you see in the strains of Magnaportha, you'll see a lot of presence-absence variation. So we have this heat map here. Let me see. Laser pointer. Yeah. So we have this kind of heat map here showing you that effectors are showing patterns of presence-absence variation across different strains of Magnaportha. So how does this actually happen? There's a lot of different hypotheses out there. One of the ones that I'm the most excited about is transposable elements. So as many of you may know, transposable elements can cause genomic changes by interrupting genes or by causing double-strand breaks or recombination in the genome that's going to cause these gene loss events potentially. Another hypothesis that we see a lot in the field is the two-speed genome hypothesis, where basically we have these compartmentalized genomes that are going to have compartments that are gene-rich, very few repeats that contain usually essential genes, and these gene-poor, repeat-rich regions that show that have a lot of effector genes. So in these plots, I'm just showing at the bottom here the distance of the nearest neighbor in the 5' and 3' of each gene. So that's the background on Magnaportha. I'm going to switch over now to extrachromosomal circular DNAs, and then we'll look back around to why I think the two might be connected. So these molecules, eccDNAs, are a very broad category. They represent very, very many different molecules, but they're all under this umbrella of circular DNAs. They can be a lot of different sizes, and they've been observed in a lot of different sizes, but they are thought to be found in all eukaryotes. How are they actually formed? Through a lot of different mechanisms. I've highlighted 3 here. They can be formed as a byproduct of double-strand breaks through the double-strand break repair machinery, as well as homologous recombination between repeats within the chromosome, and as a byproduct of LTR retrotransposon activity. So what do these actually do? they've been studied in a lot of model species, but I'll show some examples in yeast today, since this is a fungal genetics conference. So what they can do, one of the things that they do that people are the most excited about is gene amplification. So there's a story in yeast where the yeast have put a general amino acid permease on a eccDNA, amplified it massively, and gained nitrogen stress through that mechanism. And in that case, the gene is actually fully excised from the genome, and it causes a genomic deletion in those yeast. Another example in yeast that I was very excited about is that these circular DNAs can also pop out of the genome and then move into different areas. So we have this biosynthetic gene cluster in these wine yeast that sort of have moved around the genome as a circular molecule. And one of the cool facts about that is that people think that this might have been horizontally transferred as a circular DNA from a different wild yeast. So a lot of potential in those molecules. And what I'm hoping that you sort of realize is that, just like I did, is that you see a lot of similarities between these two topics, where we have circular DNAs forming in repeat-rich regions of the genome and causing these gene deletions or genomic rearrangements, and those themes kind of showing up in fungal plant pathogen evolution, genome evolution as well. So we set out to answer the question, like maybe eccDNAs are contributing to this rapid evolution in Magnaportha oryzae. So I'll talk about some of the methods that we used really quickly. Basically, the protocol to extract these is actually fairly simple. It uses mostly two enzymes. We have this exonuclease that's going to digest linear DNA from our total genomic DNA, and then a Phi29 polymerase that's going to amplify the remaining circular DNA in the cell. And then we submit it for Illumina paired end sequencing. Then we do a reference-based eccDNA calling. And to do this, we're using two different kinds of evidence. These split reads that will come from the circularization junctions here, split mapping reads, as well as paired end reads that are going to be facing the opposite direction from each other. And you see here some evidence that I've combined here, the split reads as well as the opposite facing reads that are supporting these eccDNAs that I'm showing in this plot too. And we do validate some of them by PCR as well to make sure that we're getting them right. So let's talk about some of the results from this analysis. So like I said, this has been studied in a lot of model organisms in the past especially. There aren't too many whole circularome data sets out there yet, but I did compare my fungus to some of the other ones that are out there. And one of the first striking things that I saw was that actually the whole genome was covered in sequencing reads. And to me that screamed, okay, there might be many of these molecules being formed in Magnaportha. And when you look actually at the total counts, this like sort of diversity of eccDNAs in the cell, we found that we had many more regions represented in each of our samples in Magnaportha than in Arabidopsis or rice or humans or yeast, up to a 10 to 100-fold difference in that case. The other thing that you might notice from these pie charts is that we found retrotransposons being enriched in those circular DNAs compared to other organisms. So like I said, I've represented this enrichment here. So we have compared to rice and Arabidopsis, which have these LTR gypsy elements and the LTR copia elements. And here I'm showing you the number of these circles that represent these retrotransposon sequences, normalized, of course, to the amount of those sequences in each of the genome. And you do see the clear enrichment of these retrotransposons on the circular DNAs. So what about the genes on eccDNAs? Obviously, that's probably what people are going to be the most excited about. So like I said, very high number, very high diversity of these circular DNAs. So it wasn't too surprising that we actually found the majority of the genes in the genome were at some point found on a circle. So here I'm showing you basically just a normalized read count that essentially represents how often each gene is found on a circular molecule in our data. You'll also notice that not every single gene is on these molecules, and we have these points down here in black that represent genes that aren't on circular molecules in our data. So let's talk about those genes first. So these are our eccDNA absent genes. So you'll find there's a very big list of these, and I encourage you to go to my preprint and check those out. But one of the first questions we asked, obviously, is is this a real result or is this just due to undersequencing? And so I did this permutation, this rarefaction analysis that basically showed that, well, it doesn't really look like it's an effect of undersequencing, actually. When I took a closer look at those genes as well and performed some gene ontology enrichment, I found that a lot of terms related to the cytoskeleton were showing up in these genes as well. So again, these are the genes that we never see on circular molecules. And the reason we were excited about that is that it's been shown in yeast that if you put actin in multiple copies within the genome or on a plasmid, the cells get really sick and die. So to me, that screamed, hey, there's like something going on that is preventing these types of genes from being on circular molecules. Basically, despite the coverage being throughout the entire genome, it's not this totally random thing. And there are genes that are actually excluded from eccDNA formation. So of course, what else? What else can I tell you about the genes that are on these circular molecules? So when we're thinking about fungal pathogens, obviously we're thinking about effectors. And we found many of these, well-characterized effectors in Magnaportha on these circular molecules. And furthermore, we also found that in general, if you looked at all predicted effectors in the genome, they were for the most part enriched on circular DNAs compared to other genes. So we're obviously very excited to see that kind of result. These analyses are quite tricky to do. And so one of the ways that we got around sort of dealing with so many different molecules in our samples is by essentially grouping our genes based off how often we found them on circular molecules in each of those samples. So I told you about the genes that are not on eccDNAs first, but we also had these eccDNA enriched genes in Magnaportha, which I've highlighted in orange here. And if you remember at the beginning of my talk, I talked a little bit about the 2-speed genome and how we have these plots showing the nearest gene on the 5' and the 3' end. And what I found was that, and it doesn't show up very well on the screen, but these eccDNA enriched genes were largely shifted towards that fast part, the gene sparse and repeat rich part of the Magnaportha genome. And finally, we also found that eccDNA associated genes are more prone to presence absence variation events than other genes, which we were also very excited to see. So I'll just jump into some summary of the results and talk about some future directions. So overall, hopefully today I've convinced you that eccDNAs are this like cool biological phenomenon that you should have on your radar. Hopefully I've shown that there is something cool going on specifically with Magnaportha as far as eccDNAs go, that they are more diverse in this fungus. I've shown you that LTR retrotransposons make up a good amount of eccDNAs in Magnaportha. I've hopefully convinced you that this formation isn't completely random in the genome. I've also shown that effectors are enriched on eccDNAs. And finally, that basically that eccDNA formation, as well as this genome evolution that I was talking about at the beginning of my talk, do tend to occur in the same genomic context in Magnaportha oryzae. So there's a lot more to this project, a lot of things that I tried, and I do encourage you to ask me any questions, but also do check out my preprint on the topic. So in the future, we've talked about a lot of different things, but some of the future experiments can involve, we're very interested in seeing how stress might affect these, or how the circularome might respond to low nitrogen, or what it might look like when Magnaportha is infecting a plant, as well as, of course, looking in other fungi and other organisms. Like I said, there's only very few that have been sequenced so far. So it'd be cool to see if what we're seeing in Magnaportha is true for other fungal pathogens, even other fungi in general. And then, of course, in the future, for now we're using this reference-based calling of eccDNA forming regions, but really in the future we would want to do some sort of long-read sequencing of eccDNA so that we can fully resolve what those molecules actually look like, because they can be quite complex. So with that, I'd like to thank everyone in the Krasileva lab, especially Ksenia, my mentor, as well as Grace, who's here today. Our lab studies many different topics, and it's just very cool to share this with everyone. And I also like to thank Ursula for helping me get the transposable element annotations set up, as well as Snigdha from the StatsQuest lab for getting MagnaPortha set up in the lab. So yeah, thanks for listening. And with that, I'm happy to answer any questions you might have. Yeah. We hope to, yeah, yeah. So I have limited data that says that these circular DNA formation events are fairly rare per cell. So something on the order on 1 every like 100 to 1,000 cells have one of these. So yeah, we might need to do a lot of sequencing to get a better idea of these for sure. Yeah. Over there.
Did you recover mitochondria?
Did we cover mitochondrial DNA? Yes. Yeah, definitely. Yeah. So that was maybe like 30% of the data, something like that. We had to filter it out. Yeah. Yeah.
Are there mutants in different DNA repair pathways and you can try to understand how these things better?
Yeah, so there's a very, very dense literature on that in humans and other models. You know, it's really all over the board. So like I said, there's a lot of different ways that these molecules are formed, and they can be formed through different DNA repair pathways. So each knockout that you make is going to have a different effect on these. So yeah, it would be really cool to get make a bunch of knockouts in MagnaPortha and see how each of them affects the circularome. Absolutely, yeah. In the back.
Yeah, so I'm going to list this, but the eccDNAs work with the atomics in the nucleus?
Yeah, so they do in yeast in some cases. We don't have much evidence of that in MagnaPortha, and this is like a very understudied part of the field. I will say, and you can check it out in the preprint, I did find an enrichment of the consensus sequence for the yeast autonomously replicating sequence on the circles, but I don't think that's strong evidence that they're necessarily replicating, but just a fun, like, cool anecdote there. Yeah. Yeah, we're able to enrich them. Yeah, so they're not being, removed absolutely immediately. Yes. Yeah, that's true. Yeah. Yes.
Did you calculate copy number of each ETC relative, for example, to mitochondrial DNA?
No, we did not do that.
That would be like cool to know how many copies of ETC you have, and you have the readout of the mitochondria.
Yeah, so the mitochondrial, we actually saw circular DNAs forming from mitochondrial DNA, so we can't exactly do like one-to-one to like whole mitochondria or mitochondrial genomes, if that's what you mean. But yeah, that's a very good idea for how to normalize that type of signal. Yeah. Mm-hmm. 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 Yeah. Yeah. Yeah, that's where I was getting that 1 in every 100 cell number. It's not published though, because, well, for a lot of reasons, but yeah, so I don't want to talk too much about that, but yes, absolutely. That's a great idea. Yeah. Yeah. Yes?
So these genes that are never in the circular DNA,
yeah.
there's a mechanism to avoid deformation, or you can simply selection?
Yeah. So it's not histone marks. It's probably not histone marks. It's probably not expression. So I see nothing particular about those genes at that level. So I think saying that it's selection is very appealing, but I don't have any direct evidence of that. But yeah, that's what I'm hoping. That'd be very cool if that's the case. Yeah, absolutely. Okay, thanks so much, everyone.